Hello, everyone. I'm Jo Nerschler from the Weissman Early Childhood Program, and this is the first in our series of our Early Childhood Seminar series, and we're really fortunate to have Leslie Seltzer be our first speaker. She, in case you don't know, she is a researcher at the Weissman Center, and her work looks at early experiences in life that are stressful. They might change the endocrine system of a young developing child. So she's going to talk more about that. And then following her talk, we'll have some question and answers or comments, if you'd like. And what I'm going to do when I sit down is to pass around a sign-in pad. So please write your name and contact information and where you're from, what program. So with that, here's Leslie.
All right, thank you. And thanks to all of you for coming. I'm really excited about being here and giving this talk today. I must have been to 100 of these in my life, maybe more. It's also nice to see so many new faces in the audience. I'm assuming it's fall, right? We have some new graduate students and potentially some Len Fellows who came in. I came here as a grad student as well in 2007. I just completed my qualifying exams in another department and started my PhD research here. And I was very fortunate, and in less than 2 years I was finished. was given a number of postdocs subsequent to that, and so today we'll be talking about the research that I've done so far. I work with human participants almost exclusively, and today I'm going to be talking right here at the podium so people who can hear me from far away. My main research interest is what I'll be discussing today, which is how the experiences that we encounter early in our lives can change our neuroendocrine or hormonal systems, especially in response to social cues. And since the PhD that I ultimately earned was in biological anthropology, I'll be talking a little bit about adaptation today as well, in addition to health in children as well. And last, I'll be talking today about some new research that was just published about an epigenetic study that my colleagues and I did. So we'll be talking about plasticity to some extent, and I'm excited to share that with you today. And so we have this intuitive understanding that childhood is a really important time in human development in terms of shaping our future adult behavior. We can all think of things like formative experiences or terrible things that happened or terrific things that happened that all went into making us who we are as an adult. But how does the social environment that we experience during childhood affect our brain and our biology as we grow up? And we know that our genes play a substantial role in that. However, our environment also play a role. Between those two things can play a role, and together they can affect our susceptibility. Plasticity, this fine-tuning of our biology to the environments in which we live, especially during our childhoods. And more specifically, for most humans, our relationships with our primary caregivers are a really important influence on who we grow up to be. So today I'm going to focus my research on my research contributions to date, which is understanding more about the biological mechanisms of stress responses and the hormonal processes that allow us to be comforted by the people that we're the closest to, normally our parents. And specifically, I'll be talking about those parent-child relationships and how social communication between parents and children can affect the developing brain. And I'll also show you some new discoveries about how children with poor relationships with their parents differ from controls in how they respond to and also in how they recover from stress. And hopefully it'll be an integration of something more biological and anthropological and sociocultural and psychological, all of which make us uniquely human. And so just to structure the talk a little bit today, I'll be starting off talking about the hormones that are involved in stress and social bonding and comforting. These are measurable in the lab from hormones that I collect non-invasively. And then next we'll be talking about just species-typical responses when typically developing children encounter a stressful life event. And then they have the opportunity of interacting with someone that they love and trust afterward. And again, that's kind of early graduate school, and we'll be moving along through my postdoctoral work after this. And then I'll also be focusing on what happens when things go wrong, what happens in atypical populations, specifically victims of child abuse. It's really important to understand why children who come from those abusive or otherwise averse early environments might look different from control populations with respect to their health or behavior or even reproductive outcomes. And again, because we're an anthropological group, we're also going to be thinking about maybe not just focusing exclusively on the downside there. Again, of course, children who come from those kinds of environments may have those adverse outcomes later on, but there may be actually an adaptive reason for why children who come from those types of environments might want to accelerate through the juvenile period and reproduce earlier, perhaps because you don't know what tomorrow may bring. And then last, or second to last, I'll be talking about that epigenetics study that I mentioned. And finally, some conclusions and future directions for research maybe that you can do someday if you're interested. Okay, so just some background. I mean, in essence, a hormone is really just any molecule produced in one part of the body that's going to have an effect in some other part of the body. And when we think about hormones, maybe we think about things like puberty, but hormones actually do lots of other things. They govern socioemotive states, for example, are really important, and they act as the proximate causes of behavior. They motivate behavior to some extent, and they drive our moment-to-moment actions as well as our long-term behavior, depending perhaps on what our early experiences are like. And the hormones on which I focus are called oxytocin, a related hormone called vasopressin, which you've probably heard of, and cortisol, which you've definitely heard of. And so we'll start with oxytocin and vasopressin. These are really important hormones. They help to regulate social behavior in lots of different species of mammals, including humans, particularly when they encounter stress, but you're probably more familiar with oxytocin with respect to its role in smooth muscle contractions, physiological reactions, lactation, sexual response. But of course, we now know that it's also involved in social bonding. And so this is a brief, just a small cartoon about how trauma or the threat of a cue, at least this is in laboratory animals, not in humans, could increase oxytocin, which then is going to fire your social reward circuit. And then that can build all of these positive relationship indicators between those two parties and ultimately lead to improved social functioning. And for years, scientists thought that oxytocin release was to a great extent contingent upon tactile contact, because they were looking at mice and they were looking at rats and they were looking at grooming behaviors. And again, they were also looking at lactation and birth and all these things that we know are related to the release of oxytocin. And indeed, these reproductive and social behaviors are some of the most ancient functions of oxytocin and the related peptide vasopressin. But as the years went by, and perhaps you're aware, it just became clear that oxytocin also has a lot of social functions too, between parents and their offspring, in ways that we'll show here today are not necessarily tactile at all. There are other ways you can release oxytocin as well. And the reason there's a gorilla on this slide, that is actually in the Central African Republic. There's a mother, her name was Mekome, and the baby was December. And the reason that I ultimately came up with this non-invasive oxytocin assay was because I started off graduate school as a non-human primate researcher. I lived out in the field and I was collecting urine samples out of primate nests, and I wanted very much to come up with a non-invasive method of looking at non-human primate behavior. I had no idea I would ever work with humans when I first started graduate school at all. And so what I wanted to do was go back to the field after developing a non-invasive way of looking at this socially really important hormone in Western lowland gorillas. And I never got back there again. I came here to UW, where I started graduate school, and started working with Tony Ziegler, who is over at the UW Primate Center. And she and I together developed a non-invasive oxytocin assay, and that was more or less right when I arrived here at UW. We really don't want to be sticking people with needles or pins, or certainly gorillas with needles or pins, to get a biosample from them. That's a great way not only to endanger yourself, but you're going to ruin what you're trying to observe. You're trying to look at social behavior, and if you're poking somebody with a pin, it's going to be a challenge to get at any kind of ecologically valid measure of what you're actually attempting to get at. to say nothing of the fact that that's extremely challenging to do in the wild. So suboptimal, and so I decided I was going to come up with a urinary assay, and so we went ahead and we invented one. And then I met Seth Pollack, who gave me a fellowship here in his lab, and he said, why don't you use your assay to look at human behavior? That would be really cool. And I said, okay, and like I said, I never got back to the jungle again. I've been here at the Weizmann Center ever since. But that really did open up the door for a lot of behavioral research to get at these fascinating questions about what motivates human behavior in the proximate or the immediate sense. And so we all kind of know what cortisol is and the HPA axis here is, and we'll be talking about that a little bit today. We'll be talking about how typically developing people in the first part of the talk recover from an acute stressor. It's really important. Animals feel stress, they release cortisol. And it's the end product of the activity of the HPA axis, the purpose of which is to help fine-tune your personal biology to your external environmental milieu, so you can respond to the things that are happening in your world appropriately. And I mean, of course, if a cortisol response goes on for too long, it can make you very ill. I mean, depending on the species, there's just a number of examples from other mammals. There's either a gnu or a wildebeest that they engage in these really intense mating competitions, and the losers of those mating competitions, these males who don't wind up getting access to females, have so much cortisol flowing through their system they actually begin to digest their own tissues, and then the lions come, and that's the end of them. In humans, though, cortisol does also have kind of a bad reputation there as well, but over the short term, in an acute sense, it's not really all that helpful to think about cortisol as being a bad thing. There's eustress too, and that's the kind of stress that helps you out and it helps you to remember. So you can take potentially an emotionally salient event that you encounter, and that can help you to remember that event, and that can help your memory to jog you along. That's why originally people started giving exams to students, right? You have a little bit of stress when you go into your exam, and that helps you to remember the information as long as it doesn't get too much, and then you can recall that information later as you need it. And cortisol is going to help you to do that, to tailor those individual experiences into memories that are going to help you later on, and maybe help you when that same circumstance comes up again. And here's an example of cortisol in action. We all know kind of how your cortisol response feels. It's not like a jolt or like an adrenaline response, right? It's a little bit slower, makes you feel maybe a little bit nauseous. It's kind of that increasing butterfly sensation when you anticipate an upcoming stressor. And this is an image of Sumaida Mulk. She comes from Arizona, and she is competing here in the semifinals of the National Spelling Bee. Has anyone ever been in a spelling bee? Raise your hand if you've been in a spelling bee. Raise your hand if you won the spelling bee. Right, yeah. And she's really, I did not win the spelling bee either, for those of you who did not. For those of you who did, awesome. But she's trying really, really hard here to remember how to spell a challenging word, and there's millions of people watching at home. This is on ESPN. Like, you can watch the National Spelling Bee and see who's making it to the finals. And her cortisol is really soaring here. And you can see it from her face, but she's not really angry, and she's not really mad, right? I don't know what you would call that facial expression. But she's kind of puzzling it out, right? She's thinking about it. She's maybe even, I don't know, she's smiling a little bit there. Like, it's an ambiguous facial expression, maybe. But in a case where you're in the spotlight, you're being evaluated, people are watching, there's a consequence to whether you're doing poorly or well, and that cortisol response may be helping her to focus and remember how to spell that word. And, oh, I saw a word that sounded like that, and it sounds like to start, but really it's a P, right? And so it's helping her potentially to focus and do her best, and in fact, she nailed it. And so hopefully her rewards circuits are also firing here as well. And that's what we would expect in a healthy child. Most children who come from these warm homes, we're going to see in the next couple of slides, you're going to see a big hormone response, cortisol response, as a consequence when a person encounters a stressor. And that's a nice, healthy response that you would like to see. And so I'll start by kind of talking about how you recover from social stress in the context of those warm social relationships, specifically the maternal-child bond. And I figured if we're going to see differences in my newly developed assay, maybe the best place to start looking would be those mother-child bonds, because again, these ancestral functions of oxytocin are really well described in female organisms. It's a really integrated and important system. And so I thought, yeah, if you're going to see that, you're probably going to see it in maternal-child pairs. So that's what I started with for my dissertation work. Human beings are a lot more complicated than just communicating socially via tactile contact. We don't just go around touching each other to convey social information. I guess some of us do sometimes, right? But a lot of the social information that we convey is conveyed verbally. And because I'm an anthropologist and I was very interested in how humans might differ from other primates, I wondered if language, which we're really very proud of, like, oh, that's really unique human behavior, and if it's at all special in terms of what it can do for comforting and facilitating bonds and how that might work, and whether we could potentially release a similar response just from a verbal cue rather than from a tactile one. And so I thought I would start there. And I hypothesized that humans might be able to use more than just this one form of contact, tactile contact, as a means of providing comfort, and that I'd be able to measure that in a laboratory environment. So for my dissertation study, I designed a test. I had 61 female kids, 7 through 12. They had to be prepubertal to participate. I was not able to use an image of any actual participants. I don't know why there's a dog in this picture. This is the Trier Social Stress Test. This is the adult version. The Trier Social Stress Test for children is a task in which you would have a 3-panel judge. They're all trained to maintain a neutral facial expression, and you stand up there in front of the audience, and you're asked by the judges to complete a series of questions that are designed to be too challenging for children in this age range to successfully complete. So there's a serial subtraction task where you're subtracting prime numbers, so the ones digits is always different, and you're not allowed to sit down, and nobody's smiling at you, and it does kick up a good cortisol response in many but not all children. And then after they were done with this task, which lasted about 15 minutes, I divided them into 3 groups. I had one where they had complete contact with their mom, by which I mean the mom was in the room, she was verbalizing, she was providing tactile contact, presumably olfactory contact, and lots of other forms of contact as well. And then there were those who had only verbal contact, so they just talked on the phone. They were nowhere near the same room. In fact, they were divided right after they had signed their consent documents. And then last, there were children who just rested by themselves and they had no form of contact whatsoever. And then after they were done, they would just watch March of the Penguins, which most children find extremely boring. And I collected just, looking back on this, I can't believe we collected so much saliva as part of this study, or so much urine for that matter. And at every 15-minute interval, and I just wanted to point out, if you're ever doing a study where you're measuring cortisol and you're going to do something in a laboratory context, you have to let your participants rest, because they show up and you're measuring cortisol from whatever happened to them 15, 30 minutes before. It's wildly different. It's highly variable. So you have to let them rest. Go ahead, and I gave them about 30 minutes of rest, and then you take your baseline sample. And then I administered the Trier Social Stress Test for children. Lasted about 15 minutes. This is the no contact condition here. That 0-minute point would be the point at which they would interact with their moms if they were in that condition. And then March of the Penguins all the way on out. Neutral film, boring film, whatever you want to call it. And we just collected, what was it, saliva? 7 saliva samples, 4 urine samples for each participant. So here are the data, and I just want to point out what's on my X and Y axes. So here's the time course on X, baseline. I didn't even include the arrival sample because it was so wildly variable and didn't seem to have anything to do with anything. And so there's your time course of the study, and there's your salivary cortisol measurement. Can you guys see the shaded bars there? Yeah? Okay, good. So the blue is the course of the stressor, and that flesh color or peach color, can you see that at all? Like blue and then pink? It's supposed to be subtle so it doesn't distract from the data. Sorry if it's not as visible as it could be. But that's the course of the maternal interaction, if any, during the course of the study. So here's our full contact condition. So here are these kids, and they were stressed out by this task, and then they were interacting with their moms, and their moms gave them a hug and said, "I'm sure you did great," and they sat for 15 minutes and interacted in a species-typical fashion. And here's our kids who had no contact. They just rested by themselves. And then there's the kids who had verbal contact with their moms just over the phone. This is just the sound of their mom's voice over the phone that creates this difference. And by the end of the study there, at the 60-minute mark, they're all the way back down to baseline again, whereas the children who had to rest by themselves and nobody was there for them were still significantly higher than that of their peers. And so that's a really novel finding, right? That's why I was able to finish my dissertation so quickly. I published this paper. And so their cortisol response seems to be buffered by the sound of their mom's voice. And it was a really novel finding that just hearing this trusted voice can reduce your stress level without any other kind of contact. And then we measured oxytocin too. Same idea. And there are the kids who have had, these are the control girls again, they had no interaction with their parents. And you can see oxytocin doesn't really change that much just in response to a stressful task. But these are the kids who had the full contact condition. And also, and this was one of those things where you're like, what am I going to find when I do the bioassay? The kids who had verbal contact had a really comparable response to those who were able to interact in person, and a very different response from those children who were simply left alone. And so the take-home point of this study is that contra earlier ideas about how you need touch to release oxytocin, in fact, it's not really true. Girls in the full contact condition do show that release of oxytocin, but you also have this completely non-tactile form of contact, namely socially supportive speech, that does the exact same thing. And what this suggests is that touch and comforting speech might be fundamentally similar in terms of what we use them for in our species, sending those signals of social support. And it works from a distance. And just as an aside, this was the first time I had ever really published a paper with respect to humans. And it was published in the Royal Society there in, I think, 2010. And the guy who is the press release person for the university contacted me and wrote up a press release. And it got just an unbelievable amount of press, and I couldn't understand why people were so interested in this. And then I remember that it was coming up on Mother's Day, and the news didn't have a lot of other things to report on. And so it was in like literally every newspaper. It was translated into 17 different languages. It was all very bizarre. And David Letterman made fun of it on his show, like he brought it up. And I was like, what is this? And then I remembered, it's Mother's Day. I had no intention of doing that. Yeah, so that was an interesting ride. But then for all my other press releases, I contacted that same guy, and we did it a bunch more times. It was fun. So to kind of conclude about the first study, urinary oxytocin is measurable. We can use it. This is a thing that you can actually do in the lab, and we helped to create this novel assay that you can use too now, and now you can measure oxytocin yourself if you're interested in doing so. Also, that you don't need tactile contact to release it. You can reduce your salivary cortisol and increase your oxytocin after a socially challenging event. I can measure it in the lab just from vocal contact. And I'm sure you can think of a lot of problems with this study too, and so did we. So the next thing that we thought about was, well, that doesn't necessarily say anything unique and interesting about humans, right? Because a speech act is composed of more than just the grammatical content of what it is that you're attempting to convey. Speech is a pretty complex thing. And so are we looking here at something that's really unique to humans? That's what my dissertation committee said. I don't know if this is unique to humans. Maybe it's something that's common to a lot of other organisms that use social vocalizations to communicate. And so we really needed to find a way to unpack that a little bit, that verbal exchange, to kind of piece it apart if we wanted to figure out what was actually conveying that signal of social support and comforting. And we wanted to make it resemble a phone conversation as much as possible. So again, just the sound of a voice, not the same thing as language. Maybe it was like a prosodic cue, like the tonality of what it is that you're saying. So if I were to say to you, that's a really great idea, it would mean the exact opposite as if I had said, that's a really great idea, right? The actual tone of your voice is going to convey meaning beyond the grammatical content of what it is that you're saying. And so maybe it was that, or maybe it's the fact that they're talking to their mom, right? Like the individual identity of the person with whom you're having a conversation. And that would be a really cool finding as well. And so there's really two ways you could have done this, come to think of it. You could either have tested girls' response to vocal tone without language, which would be really hard to envision an experimental condition where you're just like vocalizing but not talking. Or you could convey it with language without sound, which happily we have text messaging to do that. So that was another thing that we added to our experimental condition to try to unpack that. And our hypothesis was that texting wouldn't really do anything different from just resting in the control condition. It's a popular method. Everybody knows how to do it. You can just do it. But is it similar to or different from speech? So same or similar experimental design, girls more or less in the same age range, did the Trier social stress test again. And this time we had 17 girls in each condition, and in addition to the previous 3, we added the text messages to see if one thing we could replicate what we found before, which was kind of cool. We wanted to see if it would work again, and then we also wanted to add the texting to see if that would work too. And here's everybody. It looks kind of similar to the first chart, so that was good. It looks pretty comparable with the data I presented earlier for the 3 conditions we already discussed, these controls, these full contact kids, and these verbal kids. And then let's look at cortisol for instant messaging or texting. We call it instant message condition, texting condition. We used them kind of interchangeably. And then when you use instant messages, and you can see that it's really not that different from just resting by yourself, right? Maybe that's why people like it. It's not a socially significant event, right? You're just kind of texting with somebody. So that's your cortisol data. Looks pretty similar to just resting by yourself. Doesn't seem to bring you back quite as quickly as the in-person interaction with mom can do. And this was a really neat finding as well. And you're going to have to look for the bar here, or the line here. And if you add it, it's just way down there. Like, you're not really seeing an oxytocin response in response to simple text messaging. It's all the way down there. And so the conclusions are pretty straightforward, I think. Texting is about as good for stress buffering or oxytocin release as just resting by yourself. Something's missing from that exchange when we talk in person or someone is physically there. But what is it really about the sound of the human voice that can do that? Again, it could be prosody. Lots of animals use prosodic cues to communicate socially relevant information, like sexual motivation, fear or pain or something like that is pretty common in non-human animals. And again, it could be the identity thing. Maybe it's mom talking. But yeah, everything that I've said up until now is kind of what you would expect from like a typically developing group of kids who have a warm and nurturing and supportive relationship with their moms. And this is just the typical way in which kids would respond to a stressful event and then be comforted by it. And, oh yeah, texting doesn't really do anything, really. Okay. So what I want to do next is talk about what happens when children don't have a warm and nurturing relationship with their parents. One of the most surprising things that's ever happened to me in my scientific career was how easy it was to get parents who abuse their children, just diagnostically or abusive parents, to come into the study and be evaluated. They would just sign up, complete the paperwork, do it, and we now have a very large database in the Child Emotion Research Lab, which is headed by Seth Pollack, of children who have been abused as kids. And so we really wanted to look at that population more closely. One of my dissertation findings that's not up here was that we had about 12 kids who I thought were in my control condition who just didn't respond to the Trier social stress test at all. They didn't have a cortisol bump. And when we looked back again at our records, it turned out that 8 of those 12 kids had been abused as children. And so when I presented those findings to Seth, he said, let's do a follow-up study and we'll look explicitly at that question. What happens when you have a disrupted early environment? And we know that abuse in children is associated with adverse psychiatric and psychological outcomes, depression, anxiety, increased risk of abusing their own children someday. Even physiological diseases like cancer and heart disease, the risk for these are increased in children who have experienced physical abuse. So it's a really important question. However, the literature here is so mixed and so contextual. You can see high cortisol in kids who have been abused, or actually in many of these studies, these are adults. Very few of these actually work with children in the laboratory, which is one of the strengths of the study. These aren't people who are recalling past abuse as adults. These are children who we can document are actually experiencing abuse now. So you're not relying on a recollection necessarily. It's demonstrably true and ongoing at the time. And as for oxytocin, again, precious few studies in humans up until that point because they were doing blood sampling in order to measure. And so again, like you kind of wonder, well, you have a blood sample and you had to stick this person to get it, so what are you even really measuring? And so we really wanted to piece that apart as well and figure out what was going on there. So for the hypothesis of the third study, we wanted to see if that finding about the cortisol response in our abused children was something that we could see in a larger sample. Wanted to see if they would show deviations from controls with regard to their hormonal processes as they responded to stress and social comforting. And so for this one, I decided to look at both male and female children. We had 73 kids aged 8 to 11. We did the trier social stress task again. And then the experimental condition was just abused or not abused, right? It was not, they weren't going to interact necessarily with their parents after the task. We just wanted to see how they were recovering from this stressful experience and what their oxytocin looked like. And then we did our salivary cortisol and urinary oxytocin. And so these are the same, this is the same exact type of chart that you saw before. And these are our boys. So we had abused male children and control male children. And they did not honestly look fundamentally distinct from one another with respect to how they responded to the task. And I'll add female children. Here are our control girls. Again, it's nice to see when you do different studies with different kids that you're still seeing the same thing. Control girls, big boost of cortisol in response to the stressful task, and then over time it kind of goes away. But then look at the abused girls when you add them to the chart. They're not really responding at all. No statistical differences between those control girls, control boys, and abused boys for most of the time points. But those girls, those female children, are showing a dramatic difference with respect to the two conditions. And that all by itself is very curious. And and we'll talk about what that means here in a moment, but with regard to oxytocin, the findings here were also really striking. These findings were published in Child Development in, what year was that? 2012? And there's our abused girls with respect to their oxytocin response. And this is just in response to the stressful task. It's just this massive, unmistakable spike. And so let's kind of figure out maybe what that means here. So just to conclude, abused boys and control boys aren't really going to differ with respect to their cortisol or oxytocin release after their social challenge. They didn't respond in terms of vasopressin either. We measured that too. However, abused girls look really, really different just with respect to both cortisol and oxytocin after that social challenge. And this was another interesting response that we got from the community. This press release was actually posted on Reddit. It was the first press release that I put there to kind of hear from people about what they thought of the study. And it apparently is the most clicked on study to ever come out of UW-Madison. Gosh, so many children are the victims of child abuse. And people just really were interested in commenting on this. When you look from an evolutionary perspective, again, like it may be advantageous for girls in a very stressful early environment, just to kind of distinguish them from boys here for a minute, to develop social relationships with people who are not in that family circle. Maybe that's kind of a motive to push that child out of the family environment and go form a social bond with someone else. We know that children in these groups do tend to reproduce on average earlier than their peers do, so maybe that's a social motivation to move on out of there. Kind of almost looks like the roles have been reversed, right? So what you would think of as being a stressful experience is actually causing more of a social bonding hormonal response. And then the opposite would be true for cortisol. So we really would like to understand a little bit better exactly what that oxytocin spike is doing for girls in this different condition. So that's one of the future directions we'll be talking about today. But if anything, it raised more questions than it answered. And so in the last section of the talk, I want to discuss some new results that came about as a consequence of, again, just following where the data will lead you. We wanted to know why those abused girls look so different from comparison girls. And so it made sense to begin an investigation as to whether or not the genes responsible for the hormones themselves are being affected. And if so, if we could measure those differences at all in a laboratory environment. So we started to look at methylation patterns. Methylation is basically something sitting on top of your sequence there, and it can reduce gene expression. Methylation is acquired when you encounter early life experiences, and you do want some of your genes to be methylated as well. If your cancer genes, things that were related to cancer, were not methylated, you would start having unregulated cell growth, and that's not good. But people do differ in terms of where on their genome certain things are being methylated versus not. And it's possible to kind of engage in sort of Lamarckian thinking here. And if you're methylating something and it's in one of your sex cells, it's possible that you could transmit an acquired character into the next generation. And so I ran essentially a GWAS study. I chose, well, I'll tell you exactly how I did this here in a minute, and we hypothesized that that the methylation patterns would differ between those kids who had been abused and those who hadn't. And further, we hypothesized that expression of genes should vary as well. It's all very well and good to say that something is methylated, like something is blocking transcription, for example. But if you're not actually showing differences in expression patterns, it's not as meaningful a result. You want to show both of those. So if you're ever trying to publish a paper about methylation, you should be looking at expression data as well. So what I did was I selected, from the same study that I reported on before, I selected the 11 most severe cases of child abuse and contacted those families again. I wrote to the IRB and I said I'd like to recontact these families to ask them if they'd like to participate in a future study here, and they said okay. And so we had 11 of those physically abused girls come back into the laboratory. And we also had 11 controls that had really, really tranquil early life experiences come back as well. And we had to gather some more saliva because, if you can believe this, we didn't have enough because we had used all that saliva from the previous study. We need to get more. Also, the kits that you would use for a gene expression analysis have a different buffer than the ones you use for a methylation kit, so you have to buy separate ones. And so we brought them back into the laboratory, and again, we selected them for these extreme cases. I'm looking for bookends in this study. And we conducted what's called the life stress interview, or inventory, depending on who you ask. This is an interview of both the parent and the child, where you ask them about their early life experiences, and you essentially rank their peer relationships, you can rank their academic performance, and then you come up with a lifetime stress score from 1 to 10, with a 1 being my goldfish died, all the way up to a 10, which is the worst possible environment in which to grow up that you could possibly imagine. And then we got those 2 kits, and we sent them in for GWAS analysis with subsequent expression analysis as well. And we also did the CBCL for this study just to make sure that those bookends were clean bookends, and indeed it looks like it was. The CBCL is a child behavior checklist, and you can look at various behaviors and patterns in children's responses. And so externalizing symptoms really look different between those 2 groups. Internalizing symptoms looked very different between those 2 groups. Things like anxiety disorder and just overall or global problems with behavior looked different between those 2 groups as well. So it looks like the bookends were clean. And then we basically looked at differentially methylated nuclei. So how do those 2 groups look different? Where do their methylation patterns look different between the high stress and the low stress children, using a really cool package called RLEMA. And we used a similar package for expression analysis. And then there are gene ontological libraries that you can consult. If anyone publishes a paper and they say, I found this gene, it seems to be doing this thing in a mouse or a rat or a human or a monkey, they'll make it basically into a library that you can search, and you can say, oh yeah, that gene is known to be associated with the following 74 outcomes in the published literature. And then you can essentially just compare your high stress group and your low stress group for both methylation and expression data and see how they overlap with what we know are associated with stress responses with respect to the genome. I'd like to apologize for this Venn diagram. It really shouldn't be as large for that thing that has 2 units in it as for 1,046 units in it. But it's a useful diagram in the sense that you can kind of see what we're talking about here. We actually identified 550 loci that were differentially methylated based on the LSI, the Youth Stress Inventory or interview score, as our explanatory variable, and that was at the 0.05 cutoff. And some of them were positive correlations, some of them were negative correlations, but there they were. There were differences there. And you can look at nearest gene relationships as well to figure out if what you're looking at is more noise or something more meaningful, which we did as well. But basically, what I want to draw attention to is the fact that we found all these genes that were differently methylated, all these genes that were differently expressed, and there was some overlap there with known stress-related genes as well. And in the intersection of all of those circles, we find 2 genes that have all of those things in common, right? These are FHL3 and NPC2. FHL3 is a member of the limb protein superfamily. And these guys function partly as co-activators of Krem and Krebs cycles, transcription factors rather, and also androgen receptors, which is intriguing. And as for NPC2, mutations here lead to cholesterol accumulation within the brain and impaired cellular function. And in recent years, we've learned that increased cholesterol in brain mitochondria can affect their function as well, leading to more sensitivity to oxidative stress and decreased rates of ATP synthesis. And put together, these data are pretty consistent with the view that mitochondrial function within the brain should be investigated as a component of mood disorders. You're probably very interested about those 10 genes that are both differently methylated and differently expressed, but we don't know anything about them. And here is what they do. So you don't have to look in the appendix, this giant appendix with all these gene functions in it. We have some mucigens. These are lubricating the reproductive lining, and it really helps your embryo implant. Maybe that's interesting and important. Some of these genes are involved in learning and memory and synaptic plasticity. Some of them convey a disease resistance. That might be really interesting as well. Some of them, like MCG12916, is just an uncharacterized protein, which means that's like the scientific equivalent of, right? We have no idea what that area does. Also things like immune responses, allergic responses. And we know that these features are, in fact, some of those things that you would identify as being potentially related to behavioral or functional differences in adults as they grow up. You might have greater susceptibility to allergens as an adult if you are an abused child, and maybe this helps to explain why. And it's very satisfying to note that oxytocin receptor was also in that large group of differently methylated genes as well. And so that was helpful to know. Also serotonin was in there, lots and lots of other things. So the study not only really highlights their genomic as well as just transcriptome differences between controlled children and those who have suffered extreme abuse, but it also really illustrates that there's just an enormous amount of work that remains to be done. It's possible that these imprints, or as my colleague on this paper, Dr. Eilish, calls them, these fossils of past stress, may continue with us throughout the course of our lifespan and affecting our metabolic processes, affecting our behavioral processes. And work like this could be important as well, just for identification of risk. So if there is some sort of salivary assay where you can spit into a tube and I can say, yes, you're at increased risk potentially for anxiety disorder, well, that might be quite useful. I mean, that's coming up in the future. However, it's something that the building blocks are being laid for now. And who knows, maybe with CRISPR and other kinds of gene editing techniques, maybe we can edit out things that aren't as helpful for us in our growth and development and leave things that are and replace the bad stuff with the good stuff. So that is a decided future direction. And so let's talk about future directions, and then I'll kind of open it up for questions. So just to conclude, these early experiences are really important. We can measure the differences in hormonal output in children who have had one type of early experience versus another. We also found that different sensory modalities can convey social information. You don't just need tactile contact to do it. And we've also shown that physical abuse or other forms of early life stress really create differences in the way that hormones function. The hormonal profiles of those kids do look quite different, and there may be even a change in the way in which our genome functions as a result of that early life stress. And so for future directions, just a couple of things to kind of get the ball rolling for question and answer, like what is the function of that peak in oxytocin in these abused girls following a stressor? What do those genes that we identified in the fourth experiment even do? And if there are racial or cultural variants here, like things that are considered appropriate in one culture versus another, like would that be an interesting question to look at with respect to development? And then the big question is whether or not these differences may be conveyed intergenerationally as well from those who have suffered abuse in one generation and then may pass that along to the next. I'd like to acknowledge Seth Pollack, of course, and all of my many undergraduates. I only mention these undergraduates because they co-published with me on a couple of papers. I'd also like to acknowledge my colleague, Reed Eilish, and his postdoc, Ligia Papale, and Andy Madrid, who assisted with the epigenetics study. And you can contact me with any questions you may have after this as well. Q&A, I guess. Does anyone have any questions? Yeah.
What do you make of the big difference between the boys and the girls?
Yeah. That's a great question. I mean, these are pre-pubertal kids. I mean, one of the reasons we selected pre-pubertal children is because estrogen varies throughout the menstrual cycle, and so unless you're going to ask somebody, when was your last period or something, it would be very hard to control for that difference. And so one of the reasons that I thought kids would be a little bit easier to work with was because I thought, well, it's just always going to be more or less the same. Why you would see those differences in kids before they begin puberty is a really great question, and I don't know why you would see that there as opposed to elsewhere. Yeah. Yeah.
What do you, I know you didn't talk a lot about the temporal proximity of the abuse to the experiment.
Right. Mm-hmm.
But do you have any anecdotal observations about that when you looked at your data? Did you see any differences? It was a long time since you, like basically, do kids recover from this kind of thing? Or did you have any observations?
Yeah, the children that we chose in that most extreme group were currently undergoing extremely stressful family circumstances. So the time difference was zero, I guess you could say. As for whether or not they would recover, gosh, I mean, I hope so, right? Like we have entire disciplines devoted to helping people who have suffered from trauma recover from that trauma. But these data don't speak to how that would work.
For the task, it seemed like you asked questions, but are you thinking of in the future including maybe like a performance-based task that is more of a physical component to see if it elicits differences between, especially I would see that also, like I suspect that there might be gender differences that emerge there too, but also in terms of like the physiological response that a performance task might have versus like an actual knowledge-based task.
Yeah, that's a cool question. So like a sports-related challenge or
I just picture athletes, like they also have these a lot of like hormones and adrenaline and things
Sure.
kicking in where I feel like that also might induce stress, but at the same time also elicit a physiological
Yeah.
response that might be different.
You should do that study. I support you, yeah. I actually used to play competitive tournament Scrabble, which is not a physical sport, but it's like there's definitely a lot of cortisol happening there. And I often wished I could just go up to people and have them sign a consent form so I could collect their saliva and differentiate between the people who won the game versus the people who lost the game. And,
Yeah, I just feel like sometimes, like, and I see like children that tend to come from these neglect-free backgrounds, like also tend to have like low academic achievement. Like there's a lot of things where I feel like this particular task might kind of tap into those like low confidence, low self-esteem type of things.
Yeah.
Whereas a performance one might be more like, okay, this isn't related to knowledge, but more just kind of, can I do this?
Yeah, and I mean that flat kind of cortisol response
That's what I mean. So
might
the
just be,
physiological one might, if you,
Right.
it's more that behavioral activation piece that I'm asking
Yeah.
you about, because if like the girls are already suppressing, maybe
Mm-hmm.
this might elicit some other type of activation that might kind of speak to those differences.
yeah, it's like kind of like the Belsky, Steinberg, and Draper
Right, like
thing.
is it
Yep.
really more of a behavioral
Right.
thing or physiological response that they're
Right.
lacking, or is it more based on that?
Yeah, that's a great question. You should do it, yeah.
I'm kind of curious about your recruitment.
Yeah, everyone asked me this question. I was just waiting for it,
So
yeah.
I just, this maybe is just like a mom question and not so much a science question. But
Yes.
if I had a child or a child in my care who was going through a stressful thing,
Yes.
I probably would not be inclined to bring them to you and then have you give them a new stressful opportunity.
You would think.
Yeah, so can you just talk through what your recruitment looks like and maybe even your IRB
Yeah.
questions as you got this?
The IRB protocol was just for general recruitment. I was not, as you know, not originally setting out to look at abuse states and differences. So I had a flyer that was IRB approved, and I put it up all over town. I put it up in supermarkets. I put it up in childcare facilities if they said it was okay. And, you know, there was this IRB approved process. And we did have some children who had been identified as being the victims of abuse from previous studies that we knew about because they had completed various questionnaires that were like, oh my gosh, like that's a high score, and that child should be in this other group. But the vast majority of these kids came from general community recruitment. And then they fill out their paperwork, and you're like, that child just scored, right? Like that's a very, very high score. You go ahead and you conduct, and there are a number of questionnaires and other instruments that you can use to determine that. And then they just go in the other group. But there was nothing special that we did. We did not put up a flyer that said, do you abuse your children? Like, come participate in a, no, of course not. It was just general community recruitment and subsequent follow-up with the appropriate study instruments. I can't believe it either. Well, well, just to follow up with that, I remember Seth saying at one point that if you, like, say that I asked you to fill out a questionnaire, and the questionnaire had a question on it, do you smoke cigarettes? Like, or rather, how many cigarettes do you smoke in a day? We'll put it that way. And if you put 0, the number is probably 0, right? You're probably just not a smoker. Whereas if you put 5, maybe you're kind of, because you know that smoking is not good for you, and you're filling out a questionnaire, and you might kind of diminish a little bit. So if it's 5, and then they actually watch you doing it, you are really smoking 10. And if you say 10, and then they actually observe you, you're actually smoking 20. And so I wonder if, just depending on what style of parenting an individual is using, certain things just feel normal to them or normative to them, and they don't hesitate to really just report it when they're asked. They might diminish it somewhat, but they don't really hesitate to say, I discipline my child by hitting them with a belt, or I discipline my child by threatening them with personal violence, or I leave my child alone in the home for days at a time while I go and do other things. So it was surprising. Yeah.
So along that line, were you mandated to report these things?
Yes.
So when did that happen, before or after the experiment?
Yeah, so if a child does disclose to you during the course of any experimental condition that something like that is going on, yes, you have to report it. But in many of those cases, we didn't know until the child had already filled out all the paperwork and the mom had filled out all the paperwork that there was anything going on. But it's kind of a legal question of what CPS would do about something like that. So you can phone it in. I believe the rule is that if there are 3 independent phone calls, they'll actually follow up with the family, and the responsibility of the study is to report it, when they see clear evidence of it. And then if there are 2 more that are already in their system, then they would investigate and look at the family. You can, by all means, follow up with that if you have more questions.
Oh, no, it's too much.
Yeah, it is. It's upsetting. Yeah, it's disturbing. Yeah.
You didn't disclose in recruitment that you didn't say anything about it.
Nothing. No.
Were all these children, did you know if some were physically abused versus emotionally abused?
There is a questionnaire that you would fill out where it asks questions exactly like the smoking question. Like, how often in the past week have you engaged in the following behaviors with your child to parent? How many times in the last year have you engaged in the following behaviors? Some of them were neglect questions, some were physical abuse questions, sexual abuse questions, verbal abuse or emotional abuse questions, so it varied, yeah.
Were you able to sort out any differences?
What I used for the variable in the genomic analysis was the LSI, or the YSI, the youth stress interview or the life stress interview. We did not use those questionnaire scores. They had to meet a questionnaire cutoff for the abuse questionnaire, but then we're also looking at LSI score. In previous studies, we found that it's a 1 to 10 scale ultimately for your lifetime stress. Anyone who's got a score of above a 3, like, you would be, concerned. And we had some kids in our sample who were 8s and 9s, so it was severe, yeah. But yeah, you can distinguish between different types of abuse in this as well, but what we were really going for was a more global, how bad are things at home, rather than a particular type. Yep. Yep. That's a terrific question as well. You would need, if you were looking at a genome-wide association, you would need a huge sample size to look at those kinds of questions, yeah. And genome-wide association studies are often limited by funding, right? Like, you can only do so many full genomes at a time for a given study.
What do you think about for, like, how this would apply to bullying kinds of contexts?
Yeah, like peer relationships.
Peer kind of driven. So this is interesting to think about sort of the peer support part being coming through even with verbal. So I'm wondering about the abusive nature, how powerful that could be in a non-caretaker or non-home environment.
Yeah, I like that question a lot, because we know that, especially as kids mature, they can stop kind of looking towards mom and dad for their social support, and they start looking towards their peers for more of their social support. So it would be very interesting to look at peers as well. And I know that those studies have been done with young adults, so college-age undergraduates. I'm sure there are studies with children as well. I know that in Seth's lab, they actually looked at peer negative feedback, so negative feedback from peers about a performance or evaluation style task. And then they looked at similar questions to these. After this study came out, they designed another study that they had pre-recorded the peers giving, moderately negative feedback about a child's performance on something that looks very much like the trier. And it did seem to function somewhat similarly, but I mean, your question is a little bit more precise than that, right? Like you want to know about bullying and can't really extrapolate, I guess, based on these data, what peer relationships would necessarily look like. But I would imagine that they would look potentially similar. Could look, yeah. Did that answer your question?
Yeah.
Not very well, but I don't have those data, yeah.
About like the proximal versus distal and how distal influences could be and still be potent in that sort of way.
Mm-hmm. Yeah. Yeah. I wish I knew. Yes.
So your methylation profiles are generated from saliva samples?
Yeah,
So
I love this question. Yeah.
probably dramatically underestimating the amount of methylation changes, experience-dependent methylation changes you would find in actual brain tissue.
Yeah.
And also genetic mosaicism, experience-dependent mosaicism, which of course you can't look at any of this in humans. You can't biopsy brains.
Well, no, the IRB might have a problem with that in a laboratory environment, yeah.
So you can only do these studies with animals, and I haven't seen much at UW so far with genetic mosaicism
Yeah.
in the brain. Do you know if there's any kind of a push here?
I would ask Reed that question. So Reed Alish, my colleague in the Department of Psychiatry, might be better able to comment on that literature than I would be. But with respect to saliva as a medium, right, for doing methylation analysis, there have been a couple papers to come out recently to show that the data that you would get from a salivary sample, it's an echo of what you would get if you were to section out the brain and various hypothalamic nuclei, for example. But there is a decent correlation between salivary levels and levels that you would get from the brain, although at a diminished concentration. But that was definitely a criticism of our reviewers. Like, how do you know you're measuring something that's meaningful on a biological? This is the question that's followed me throughout my life. Like, how do you know that oxytocin in urine is actually reflecting a central process? How do you know that methylation data that you're getting from saliva is reflecting a central process? And all you can do is look at the literature in those cases and start to create some hypotheses about how those two things can necessarily be related to one another. And then the work does pass on to the mice people or the rat people to demonstrate the exact physiological pathways that lead to that outcome. Great question.
Okay.
All right.
Thank you.
Thank you very much.